Hello and welcome to this revision video on Biology Paper 2. This video will cover topics 5, 6 and 7, which you can have a look at in your revision guide as well to prepare you. It's suitable for combined and separate scientists. This video only contains combined content and later on I'll do a separate only video containing separate content. If you'd like to download some handouts to make notes as you go along to the video, you can do so by visiting gcserevisionmonkey.com. The kind of topics that we'll first of all look at in topic 5 are all to do with homeostasis and response. So things like hormones and nerves, controlling fertility and the menstrual cycle. First of all, we'll look at homeostasis. What homeostasis means is maintaining a constant internal environment inside an organism. So the main things that you want to control are things like your blood glucose levels. If they get too low or too high, that can cause serious health problems. You want to control your temperature within your body. Within human bodies, the temperature is controlled very close to 37 degrees C, and again, any difference around this number can cause serious health problems. And finally, your water levels. The way we control these go into more detail in the separate course, which I'll do in a separate video, but we will look at how blood glucose is controlled by insulin a little bit later on. The nervous system is one way that we can help to maintain our constant internal conditions. And the main pathway for a nerve response starts off with a receptor cell of some kind. This electrical signal is passed on from a receptor cell to a sensory neuron. This message is then given to a coordinator, which in many cases is the central nervous system that comprises of the brain and the spinal cord. This then gives the message to a motor neuron, and this finally gives the message to the effector, which will bring about the response. So, for example, if your receptor cells detect a change in temperature, for example, it can send a message to the sensory neuron, which can send a message to the brain and the spinal cord. The motor neuron will coordinate the response, and the effector could be a muscle to help your hairs on your arm stand on end, or in a different pathway it could be a gland, which will then perhaps secrete a hormone or something like that. So the effectors themselves are either muscles or glands. So this is a pathway that you will have to remember off by heart. The thing about nerve responses is that they are very quick. They act in a very precise area, and they only act for a short time. One very special nerve response is a reflex response. This response is completely automatic, and what that means, it does not involve the brain. So, for example, if this person experienced a bee sting on their thumb here, their receptor cells would detect that pain from the bee sting. The message will be sent along a sensory neuron, but this time, rather than going up to the brain, that's far too long for the message to travel because we want a really automatic, quick response. This is then passed across a little relay neuron, then back down the motor neuron to the effector, which in this case is the muscle, and that will cause your arm to move away from the sting. So this is not a conscious response. You don't think about it. It happens really rapidly and really quickly. There's another word here that I want to discuss, and that's the synapse. The synapse is the gap between two neurons. So if we have two neurons here, an electrical message is sent along the neuron, but then this is transferred to a chemical message which helps the signal jump across the gap, and then you get an electrical message the other side as well. So a connection that had lots of synapses would slow the response down a little bit as well. Another reflex response is the eye blink as well. Sometimes if you get an object close to your eye, you will blink automatically without thinking about it, and ultimately these reflex responses are to protect you. Moving on now to a required practical. There's not many required practicals in paper 2, but this is one that you need to know about. This one is all to do with measuring reaction time, which is the time it takes to respond to a stimulus. A stimulus is any change in the environment. Okay, so it could be that you notice something coming towards you, it could be a temperature change, anything that's changing in the environment you can call a stimulus. So you often change one factor when you're measuring reaction time in this experiment. So for example, you could measure your reaction time before and after something that's going to affect it, like caffeine. And in this case, you place a ruler at a certain height above someone's hand. So this is person 1, this is person 2. Person 1 will drop the ruler at an unknown time, and person 2 will try and catch the ruler. The distance that the ruler moves through the hand, measured by the top of the thumb there, will give you an indication of reaction time. Obviously, the shorter distance means a faster reaction time. So important things then, if we're looking at the effect of caffeine, we need to make sure that we use the same person before and after, that person uses the same hand, because there might be differences in reaction time if you're left-handed or right-handed, and also that the ruler is dropped from the same height. Okay, so just be aware for this one, for a required practical, you should be able to write a method, talk about variables, especially in the important things that you would want to control. Moving away from hormones then, but still something that is involved in homeostasis and maintaining the conditions within inside our body, and that is the endocrine system. It's a really difficult word just to describe glands that produce hormones. Controlling our body by hormones is slower than that of nerves, than a nerve response. It acts in a more general way. And usually the response lasts longer. Now there are several endocrine glands that you need to be aware of. The most important one here in your head is called the pituitary gland, and that has got a little bit of a nickname called the master gland, because that is often responsible for switching on and off other endocrine glands in your body. The one in your throat here is called the thyroid gland. That produces a hormone called thyroxine. The pituitary gland produces lots of hormones that we'll talk about later as well. The adrenal glands here, they produce adrenaline. You've got the pancreas here, a really important gland that produces lots of different hormones, and we'll talk about the pancreas later on too. And depending on whether you're male or female, females have ovaries, and we'll talk about those when we talk about the menstrual cycle, and the men have testes, and the hormone that's produced there is testosterone. We're going to look now at the pancreas in a little bit more detail, because we're going to talk about one of the things that's important in homeostasis, which is controlling the blood sugar levels. So the pancreas is involved in controlling blood sugar levels because it produces a hormone called insulin. It's something that your bodies will produce all of the time, and what the insulin does is, if there's too much glucose in your blood, I'll just represent the glucose by a G. If there's too much glucose in your blood, the pancreas will start producing more insulin, and that will cause the glucose to go into your muscle and your liver cells. And here it can be stored as glycogen. So the liver itself stores glucose as glycogen. So difficult words here. Glucose and glycogen sound very similar, but if the blood glucose levels are too high, it is controlled by this hormone insulin. This next bit is for higher tier only, just this slide here, because you also need to know what happens when the blood glucose levels get too low. The pancreas produces another hormone, this time called glucagon. Again, annoyingly very similar to the words glycagon and glucose that we looked at earlier. So glucagon is a hormone produced in the pancreas, and what that will do is it will cause the liver to turn that glycogen into glucose again, which will then go back into our blood. So it does the opposite of the process that insulin does that we talked about before. So higher tier only need to know about glucagon. If the blood glucose levels are too low, glucagon helps convert glycogen into glucose, which is put back into the blood. This is an example of a negative feedback cycle. So if your blood sugar levels get high and then low and then high again, your main goal is to keep them at as close to their optimum level as you can. So if the glucose levels get too high, your body will produce insulin and make them go low again. If the glucose levels get too low, your body will produce glucagon and therefore increase the blood glucose levels again, and it kind of wavers around this optimum midpoint. The trouble with controlling blood glucose, this is now for foundation and higher again, and the trouble with controlling blood glucose is it is linked to a disorder called diabetes. There are two types. Type 1 diabetes is the type that is inherited, and in this type of diabetes, your body produces little or no insulin. And this is the type of diabetes that the doctor will prescribe insulin therapy, so that involves insulin injections. And you have to keep a close monitor of the glucose intake that you're taking into your body. If your blood glucose gets really low, you will quickly eat some biscuits or some foods that are high in glucose. And you also need to regularly remember to go and get your insulin injections. Type 2 diabetes, however, is caused by lifestyle factors. It's often linked to diet and obesity, and you get that later on in life. And it's when you still produce insulin, but your body is resistant to your own insulin. So basically it becomes no longer is effective. So you're still producing it, but it's not working anymore. It's not reducing your blood sugars to normal. So your doctor will prescribe, in this case, a carbohydrate controlled diet. Because, remembering back from topics on diet, the carbohydrates in paper 1 can be broken down into sugars like glucose. So we need to control the amount of carbohydrates we're taking into our diet, and also he or she will suggest an exercise routine as well to help maintain your weight, which is obviously a risk factor in terms of diabetes. Still on the theme of hormones, we're going to look at hormones that are involved in the menstrual cycle now. So here we still have the endocrine system on the right hand side here. At the top we have the pituitary gland, and this produces follicle stimulating hormone, which is FSH, and it also produces luteinizing hormone, LH. You need to know what these two do. This follicle stimulating hormone causes the egg to mature, and the luteinizing hormone here, the LH, causes the egg to release. The other endocrine glands involved in this process are the ovaries, and they produce two hormones called estrogen and progesterone, and these two are involved in growing and maintaining the uterus lining. So the uterus of a woman will be just below where the ovaries are. The ovaries will link to the uterus via an oviduct, and this is where, if the woman becomes pregnant, a baby will develop. So if we look at the uterus lining, it links into the menstrual cycle, because the menstrual cycle is a 28-day cycle. If I draw this diagram as if it's the width of the uterus, first of all, in the first few days, 4 to 5 days of the cycle, the uterus lining breaks down, and at this point it will then be your menstruation period. Okay, it's shortened to the period, so that's your period of menstruation, when the woman will bleed and the uterus lining will break down. The uterus lining will then build up again with the help of estrogen and progesterone, and then will be maintained. Day 14 is an important day because that is the day in which the egg is released. And then the cycle will all happen again, and the uterus lining will start to break down, etc. That's about the detail that you need to know for foundation tier. For higher tier, you need to know how these interact. So if you're doing foundation tier, feel free to skip on to the next slide. For higher tier, you need to know that when FSH is produced, not only does it cause the egg to mature, but it also stimulates estrogen production. So we said before that the pituitary gland was a master gland. Well, in this way, the FSH travels from the pituitary gland to the ovaries. It will cause an egg to mature, and it will also then stimulate the ovaries to start producing estrogen. What happens then, however, is the estrogen that's produced from the ovaries will travel and signal to the pituitary gland, and that will do two things. The estrogen inhibits the FSH, and it will also stimulate LH, which is going to then cause the release of the egg. The other connection you need to know is that progesterone, which is involved in maintaining the uterus lining, also inhibits FSH, and LH as well. So at this point here, the estrogen does not is stopping further eggs from maturing. So each month you only want one egg maturing. So what that estrogen is doing is stopping more eggs maturing and then causing that one egg to be released. Next, we're going to move on to controlling fertility. So this is for foundation and higher again. Fertility is all to do with your chances of getting pregnant. So there's two things we need to look at. We need to look at things that we will do to control our fertility, to reduce it, and also for higher tier we'll need to talk about increasing our fertility through IVF. First of all then, controlling our fertility, trying to reduce our chances of getting pregnant, we use contraception. There are two main types, ones that include hormones, which are called hormonal methods, and ones without hormones, which are non-hormonal methods. So hormonal methods, first of all, there's various forms of them. All of them have certain side effects, so often people need to find out the one that works best for them. But the pill, the patch, the implant, or the injection all contain estrogen or progesterone. So you can get progesterone only pills if you're finding that side effects are being caused by the presence of estrogen, and so on and so forth. So the estrogen inhibits, which means stops, FSH. So if you remember before, FSH causes the egg to mature. So if you take a pill every day containing estrogen, you're going to stop your eggs maturing. You'll still go through your cycle and have a period, but there won't be a mature egg there. And progesterone causes a thick mucus, and that prevents the sperm from reaching the egg, so again protects you from becoming pregnant. So it's really effective, but there are side effects that go along with these. And there is also a T-shaped device called an intrauterine device. That is like they sometimes call that a coil that's inserted into the uterus. That can be hormonal, or it can also be non-hormonal as well. So it can be put over into both sections. This one will release hormones, this one is made of copper and will just kill any sperm that go into the uterus. There are also barrier methods, so physical barriers to stop the sperm reaching the egg, and they include condoms and diaphragms. And you sometimes would want to use those with a spermicidal agent. That just increases your chances of not getting pregnant and just adds that extra layer of protection. So a cream that you can use along with it that will kill any sperm that might be present. And you can also choose to abstain. So abstaining means that you are not going to have sexual intercourse on the days that you are likely to get pregnant. So around the weeks of day 14, when the egg is released, you're going to not have sexual intercourse to prevent any sperm from reaching the egg. The alternative one you can have is sterilisation. That is a permanent procedure, it's an operation, and that will involve either the man or the woman undergoing surgery to ensure that they cannot have any children. This next little bit on IVF is higher tier only, so if you are doing foundation, move on to the next part of the video. IVF is a way of increasing your fertility, and it is quite an intensive process and an expensive process, but it increases a chance of them getting pregnant. So what would happen first of all is the woman would inject herself with FSH and LH, remembering these are the two hormones involved in FSH maturing the egg and LH releasing the egg. So the purpose of this is so that the woman will generate multiple eggs, and then those eggs are taken from the woman, and the man will provide a sample of sperm, and in a lab the sperm is injected into the healthiest looking eggs. So they will look at the sperm and they will look at the eggs, and they will put the healthiest specimens of sperm and egg together. So the sperm, once it's injected into the egg, will make a fertilised egg, and if that's successful, that will start dividing, and a few balls of cells together is called an embryo. That embryo is then implanted in the uterus of the woman, and with IVF you can have 1 or 2 embryos implanted to increase your chances of success. There are downsides, including multiple births, which can cause problems for both the mother and the baby. Sometimes it's not successful, and in fact quite a few times IVF is not successful, so you've paid thousands of pounds and don't have a baby at the end of the procedure. It's very stressful on both the mother and the father, especially the mother with the injections of all the hormones. But the ultimate goal and the ultimate advantage is that people that find it hard to have children can do so. This next topic is higher tier only as well, and it's focusing on 2 hormones called adrenaline and thyroxine. Adrenaline is produced in the adrenal glands, and it is responsible for your response to fear and stress. So in situations of fear and stress, adrenaline is released. You need to know that the release of the adrenaline causes an increase in oxygen and glucose to your cells, particularly in the brain and your muscles. Brain so that you can think and coordinate your response, and the muscles so that you can move quickly and react. It will cause an increase in heart rate, and it will therefore trigger a fight or flight response, which means either you are going to fight in response to that fear or stress situation, or you are going to have the opportunity to run and flee from that situation. I would suggest learning this response, because this could be a higher tier style question in the exam. The other one that you need to know is thyroxine. This is produced in the thyroid gland, and this is responsible for your resting metabolic rate, which is called your basal metabolic rate. So this is the metabolic rate whilst you're at rest, and thyroxine is responsible for keeping that the same. It is also controlled by a hormone in the pituitary gland called the thyroid stimulating hormone, and it's involved in a negative feedback cycle. If you look at the thyroxine levels in blood, they will go up and down, but these levels are controlled by the presence of TSH. So if the thyroxine levels get too high, that TSH will be inhibited and stopped. If the thyroxine levels in the blood drop too low, that TSH in the pituitary gland will be stimulated and start being produced to stimulate the production of thyroxine. Okay, the next topic we're moving on to now for foundation and higher is all about inheritance, variation and evolution, and we'll be talking about DNA, reproduction, chromosomes, genetic engineering and all of the above. First of all then, talking about DNA and genes, you need to know that DNA is deoxyribonucleic acid. It looks a little bit like this in that it is a polymer, which means a long molecule. It is a DNA double helix, and it is found in the nucleus of your cells. Often in the nucleus, if we just take a picture of what the nucleus looks like, often in the nucleus it is just loose DNA because it's being used. It's being used to make proteins. But sometimes it can be organised in chromosomes, and you might see chromosomes like that. Also, you may also see chromosomes that look a bit more like a cross when they have doubled up and got exact copies of each other. So be aware that you might see both of those in the exam. Anyway, in the cell you have 23 pairs of chromosomes, so 46 in total. They're in pairs, they're in pairs because you have one from your mother and one from your father. So you have 46 chromosomes in total. A length of a particular sequence of DNA is called a gene, and that gene is used to make amino acids, of which there are about 20 or so of them, and a particular sequence of amino acids is used to make proteins. Your entire genome, the entire genetic information, in an organism, whether it's a plant or an animal, is called a genome. And that's really important to help us investigate inherited disorders or the things like human evolution and how we're all related to each other and how we've migrated in the past. Next, we'll talk about reproduction. The first one that we're going to talk about is sexual reproduction. So within plants, that involves the sex cells called the gametes. Sorry, with animals and plants, that's called the gametes, which are the sex cells. In animals, this is the sperm and the egg. And in plants, this is pollen and the egg. So in fertilisation, in sexual reproduction, you go through a process called fertilisation. And in this process, if we take the sperm and the egg as the example, the sperm with 23 chromosomes will insert itself into an egg which also has 23 chromosomes, and then together they will make a fertilised egg with 46 chromosomes in, 23 pairs. And then, like it says here, after fertilisation, that cell will then start to divide, and again until they make an embryo and then a full organism. And that cell division is mitosis. Next, we're going to talk about meiosis, which is a different type of cell division that happens in the ovaries and the testes. Meiosis is the process by which gametes are produced. So it happens in the reproductive organs, ovaries and testes. One way to remember it is meiosis happens in my ovaries and mitosis happens in my toe. So meiosis is involved in producing the sex cells, so the egg and the sperm, and then mitosis is involved in any sort of growth and repair in the body and cell replication after fertilisation. So what happens in meiosis is that when a cell starts to divide, its DNA will be packaged into chromosomes, and each chromosome will double up so it has two arms, and it will look a little something like this. Then what happens is chromosomes pair 1 to 23 will line up in the middle. I won't have space to draw 23, but you'll get the idea. And a first cell division occurs. In that first cell division, the chromosomes are pulled apart. But at this stage, when they're meeting each other in the middle, there's a bit of crossover of genetic information. So the genetic information that was from the mother and the father mix, and that causes variation, which is important for the end. So there's a little bit of mixing going on there. Then these two chromosomes are pulled apart. So you end up with two cells in a first division with 23 chromosomes. They still look like these X shapes at the moment, all the way down to pair 23, and the same on this side. And again, they line up in the middle of the cell. You then get a second cell division, whereby each arm of the chromosome is pulled apart, and these aren't identical anymore because of the mixing that's happened up there. So they pull apart, and you end up with cells here with 23 chromosomes in it, all the way down to chromosome 23. So I haven't got time to draw all 23, but you get the idea. Each of these at the end will have 23 chromosomes in. Each of them will be different because of the variation at the top, so these will be genetically different, and they will be either sperm if they've been made in the testes, or they will be eggs. So that's how egg cells are produced. The other form of reproduction that you might see doesn't involve meiosis and making egg cells at all. It doesn't involve fertilization because it is simply one cell making a clone of itself. That cell, or all those cells and an entire organism can make a clone. This can happen in plants or animals, and with asexual reproduction, the offspring are genetically identical. So within the animal kingdom, sexual reproduction is far more common than asexual, but within plants it's very common to see both types quite often. Sex chromosomes is what we're going to talk about next. The 23rd chromosome is called the sex chromosome. So the 23rd pair. You can either have a pair of XX chromosomes, or you can have a pair of XY chromosomes. If you have XX, you are a female. If you have XY, you are a male. And you can do a genetic cross to show that whenever a male and female reproduce, they have 50% chance of having a boy and 50% chance of having a girl. Because if we took the possible gametes from a man, we'd have XY. The possible gametes from a female, we'd have XX. And you will have to complete these crosses in the exam. So here we'd have an XX, here we'd have XY, XX and XY. So you'd finish that off by showing that it's got a 50% chance of getting a boy or a girl. Here we've got 2XX, so 50% chance of it being female, and here we have 2XY, showing that there's 50% chance of getting a male. Keeping with the theme of genetic diagrams then, we are going to draw some more genetic diagrams now, but to be able to do that we need to know a few key terms. The first one is allele. Allele is a form of a gene, or one form of a gene. Try not to say type. A type of gene you will not get the mark for. Say one form of a gene. So a dominant allele means that a characteristic is always expressed if one allele is dominant. So if you have a dominant allele, a dominant form of a gene in your DNA, you will express that particular characteristic. Whereas a recessive allele, the characteristic is only expressed if both alleles are recessive. So what that means is to express something that is recessive, you have to have a recessive allele from both your mother and your father to have the chance of expressing that particular characteristic. A phenotype is what you look like, so it's the characteristic that is expressed. So if they ask you about the phenotype, you might be talking about brown hair, for example, or tongue rolling. It's whatever characteristic you're talking about in the question. A genotype is the alleles present at the genetic level. So your genotype, for example, would be, if we're looking at tongue rolling, it could be T. T, two capital T's. I forgot to mention up here that when we express alleles as letters, a dominant allele is a capital letter and a recessive allele is a lowercase letter. So here we've got TT. This would be called homozygous dominant. Homo meaning the same, we have two of the same dominant alleles. Lowercase t's would be called homozygous recessive. Homo meaning the same, and two recessive alleles because you've got two lowercase letters. And the final one that you can have is one of each, which is heterozygous. So that's going to be important for us to draw our genetic crosses. We'll start here by this example. If you're doing higher tier, you would have to draw the genetic cross from scratch. If you do foundation tier, you'll probably be given the genetic cross and have to complete it. So this question says the ability to roll your tongue is caused by a dominant allele. What is the probability that a woman who is homozygous recessive for tongue rolling has offspring that can roll their tongues if she has children with a man who is heterozygous? So let's use the letter T again. We said recessive would be lowercase t, because homozygous recessive. Heterozygous would be T, T. So we'd cross those in a genetic cross. Capital T, lowercase t, crossed with TT. You'd have to do that from scratch if you're doing higher tier. Foundation, you would have to complete this cross here. So all you do is match up the letters. Here you would get TT. Here you get two lowercase t's, two lowercase t's, and here TT. So if tongue rolling is caused by a dominant allele, anytime you've got a capital letter, you will be a tongue roller. So here we finish it off by saying you've got a 50% chance of tongue rolling. Now we're going to move on to look at inherited disorders, and two that we'll look at are polydactyly and cystic fibrosis. Polydactyly is a disorder caused by a dominant allele, but this allele is quite rare in the population. So even though it's dominant, not many people possess this allele, but if they do pass it to the next generation, then they will be born with an extra digit on their feet or on their hands. This polydactyly is quite easy to deal with. It's dealt with at birth and the extra digit is removed, and lots of people don't even realise that they suffered with that disorder from birth. This other disorder is cystic fibrosis. This is caused by a recessive allele, and this affects the cell membranes. With cystic fibrosis, you can see here a sufferer having lung difficulties. It causes mucus and excess mucus to be present in the airways, in the air passages, because of this disorder with the cell membranes, and what it causes is really severe breathing difficulties, and people find it difficult to get enough oxygen into their lungs, and it can quite severely affect their lives in some cases. So one suggestion to help improve for such inherited disorders is to do something called embryo screening, which is looking at the embryo, which is a ball of cells right at the beginning of life, and selecting a cell from that embryo and looking into its DNA to see if it has genes which could cause some of these genetic disorders. And what they are looking into is perhaps being able to change or remove a certain gene that they find that's causing a disorder or could cause a disorder, or perhaps even suggesting that they wouldn't use this embryo and they would choose another one that the mother and father had produced instead. Obviously, that has a lot of ethical and economical and moral advantages and disadvantages behind it, which I suggest you look further into. Main advantages are that it could eradicate some of these genetic disorders, but obviously there's a lot of debate surrounding it because you're choosing what embryos survive and live, and you're perhaps prejudicing people with these disorders, so there's lots of continuing debate around it. Moving on now, we're going to talk about variation and evolution. Variation just really means that everybody is a little bit different. Not only animals, but plants and fungi, every living organism is a little bit different. So humans are a little bit different. They can be different at the genetic level, for example, in terms of things like eye colour. Those differences are controlled by genetics, hair colour, like we said before, whether you can roll your tongue or not. Environmental variation can be brought about by people having tattoos and piercings. And some variation can be determined by both genetics and environment, things like weight. Obviously, you can have metabolism and things which are genetically based, linking into determining your weight, but also what you eat in your diet and your lifestyle. Again, with height, your genetics will predetermine your maximum height, but the food that you eat and how well nourished you are will determine whether you reach that maximum. So mutations in the DNA can lead to variation. Okay, so slight changes in your genetic code can cause variations in the way you look. Many of those mutations will be only very slight and perhaps unnoticeable, but some of them, as we said before, can lead to genetic disorders as well. Mutations in your DNA will happen randomly, and many of them will not cause a problem, but some of them do. Variation then can lead to evolution and speciation. Now, evolution was a theory come up with by Charles Darwin, and his theory was evolution by natural selection. And what it starts off with is the idea that naturally there is variation within a population. For example, if you had two elephants, naturally some might have short hair, and others, due to genetic mutations, might have slightly longer hair than others. And ultimately, those best suited or best adapted to the environment will survive. Those ones that are best adapted will reproduce, and they will pass on their genes to the next generation. So, for example, in this rather far-fetched example, you could have an elephant here with longer hair, and perhaps if it got really hot and sunny in the climate, it would become a disadvantage to have long hair and an advantage to have short hair. This individual here would have a better chance of reproducing because it is better adapted to the environment, it can cool down easier, and that is more likely to reproduce and pass on their genes to the next generation. This can also bring about speciation as well. So, for example, if you had organisms that were dissected by a mountain range, if the weather conditions and climate conditions were slightly different, this isolation can cause speciation in different species. So these could be have different advantages in their environment to the organisms on this side, and if they are isolated, they can no longer reproduce and they can become a separate species. And a species are organisms that cannot reproduce to produce fertile young. Okay, so if they make babies and those babies themselves aren't fertile, then we can't say that they are the same species. So to be a species, you have to reproduce and produce fertile young. This isolation brings about speciation. One of the pieces of evidence we have for evolution is fossils. So for example, here when we looked at fossil records from horses that we had millions of years ago, we found that slight changes in their fossil bones provided evidence for how those horses evolved, and obviously newer technology in looking into DNA will give us more information as well. There are several types of fossils that can exist. You can get very simple imprints or casts of animals, so footprints or casts of maybe plants or animals where they have been. You can have rock-like fossils where animals are replaced by minerals. So in this process, for example, if an animal dies on the bottom of the sea or the bottom of a river, that animal will gradually decay and wear away. The soft body will be eaten, but the bones and things are a little bit harder to decay. So what will happen is they'll stay there for several thousands of years, but gradually the water from above will go into the decaying animal and it will replace the bones with minerals, causing a rock-like shape in the form that the animal was in when it died. The other type of fossils are fossils of animals that have been preserved. So for example, if you find animals in conditions where they couldn't decay, so where they've got no oxygen or little oxygen and moisture, for example, they will not decay very well. So for example, when they found remains of animals deep under ice, where it's really cold, it's not warm enough for decay, there's not enough oxygen, and also where they found animals in fossils such as amber fossils that have been completely preserved. Extinction next. Animals that are not best adapted to their environment can become extinct. There's several reasons that can cause extinction. An extinction is when there are no individuals of a species living. This can be caused by several factors, including perhaps a new predator that has come into the habitat, a new disease. This predator, could also involve hunting, so human hunting has brought about the extinction of many animals. Environmental change, so global warming, if the temperature changes. Competition, if they can't compete with a new species that arrives in the area, they'll be outcompeted and can become extinct. And also some sort of catastrophic event like a meteoroid or something like that, or a really catastrophic weather event that could cause the extinction of a species. Antibiotic resistance is an unusual topic within this larger topic. However, it is in this section because it is another example of how we can provide evidence for evolution. Because bacteria so quickly divide, they can evolve really quickly, unlike other organisms. So for example, within this bacteria here, this one here has a random mutation in its DNA, and that just happens to make it resistant to what we use to kill the bacteria, which is antibiotics. So DNA mutations can randomly occur in bacteria as well. If we were to treat this sample with antibiotics, these ones would die out because they don't have resistance to antibiotics, and this would then leave this individual to go on and very rapidly replicate every 20 minutes or so until you get a large population of bacteria that are resistant to antibiotics. One example of this is MRSA, and doctors are constantly battling with this bacteria to try and produce new and better antibiotics to treat it because it has caused massive problems because people are no longer finding effective antibiotics to kill the bacteria, and a lot of people are suffering, and some people are even dying because they can't have the MRSA treated. The main problems driving this are linked to the advert in the press about overuse of antibiotics by doctors. So they can only kill bacteria, so they shouldn't ever be prescribed for things such as viruses. And also the use of antibiotics in farming as well. The main thing with antibiotics is you must take your full course. You must ensure that you kill all the bacteria before you have chance that some of them might start mutating and becoming resistant. So it's really important that you do that. This also provides evidence for evolution because you can see how quickly these bacteria are evolving, and you look at the population, and this mutation and change and the change in the population is evidence that evolution is occurring. Classification then is the next thing that we're going to talk about. The first system is actually called the Linnean System. It was, however, developed by someone called Carl Linnaeus in the 1700s, and it is a system that helps us classify and sort all of the living organisms on the earth. And it starts off with kingdom, phylum, class, order, family, genus, and species. And this leads to a binomial naming system whereby every single organism on the planet can be named by two words. For example, the barn owl is called Tyto alba, and the brown long-eared bat, for example, is called Placotus auratus. And every single organism on Earth is written as a binomial system. Bi, like bicycle, meaning it's made up of two parts, is made up of its genus and its species. So what Carl Linnaeus did is he looked at all the characteristics on the animals, things like whether they had a backbone, things like whether they produce milk. However, there is an improved system which was developed a lot later in 1990. In this system is called the three-domain system, and essentially what it did was put three extra groupings on top, an extra grouping on top of the Linnean system with three domains. So the domains are called archaea, bacteria, and eukaryota. The eukaryota contains all the eukaryotes. The bacteria and the archaea are very similar to each other. They're both prokaryotes. The archaea contains primitive organisms, like those ones that live in hydrothermal vents and places such as that, so those kind of extremophiles. Then we've got the bacteria and the eukaryota, and then underpinning that you would then order them into kingdom, phylum, class, order, etc., all the way up to genus and species. Now, the reason this three-domain system was proposed was because there was better technology, much improved microscopes. They could start looking inside cells, comparing the organisms at a cellular level. Did they have a nucleus in them? Did they have mitochondria, for example? What kind of biochemical processes did they do? Were they respiring? Were they photosynthesizing? So with this better technology, with these better microscopes, these electron microscopes as well later on, they were looking at these at a subcellular level, and then that brought about this three-domain system, which then has the Linnean system after it as well. Way that organisms can be regrouped together as well is seen in evolutionary trees. So those animals that are most closely related are those closest together in terms of branches. So A is much more closely related to B than it is to C, because these two are very close together. C and D are really closely related, so are F and G. And then you can see them sharing a common ancestor here, perhaps thousands of years ago, and they've branched out like this. So this is constantly changing as information about the structure and the function of the organisms is improved. Okay, we're just going to discuss two more things, selective breeding and then genetic engineering. Selective breeding, first of all, is something that we've been doing for thousands of years to produce dogs, for example, and the variety of dogs that we see today. It is a way in which humans can influence evolution. We can do this with plants or animals, and the things that we do would we pick the organism, so the plant or animals, with the desired characteristic and we'd breed them. For example, if you had a variety of different strawberry plants, some were producing very large strawberries, some were producing very small strawberries. What you would do to get the biggest yield is you would select the strawberry plants that were producing the biggest strawberries, breed those together, and continue to do that in many, many steps of breeding until you got plants that were constantly breeding large strawberries. The same with the dogs, for example. You would perhaps, if you wanted a dog with curly hair, you would take two dogs with the curliest hair, breed those together, and hopefully end up with a dog with your desired characteristics. If you wanted perhaps intelligence in a dog, you can do the same thing. But the trouble is with this, it reduces the gene pool, which means it reduces variation, and especially within the dogs, it can cause lots of breeding problems and fertility problems and things like that. The last thing that we're going to look at is genetic engineering. In this process, really for foundation, all that you need to know is that a gene is cut out of one organism and inserted in another. For example, you might want to cut out a gene for drought resistance and put it into a wheat plant, for example, to get a really good crop of wheat. So you would cut out this gene from one organism and put it in the wheat plant. You might want to cut out a drug for pest resistance, for example, or in some cases you might want to cut out the gene for insulin and put it into bacteria so that bacteria produces insulin. In a bit more detail for higher tier, this is the exact process that's going on. You're taking the gene from the cell that you want, you're cutting out your desired gene. So for example, this could be the gene to produce insulin to help people with diabetes. You would then cut out a DNA vector really what's normally used is a plasmid from a bacterium, that will then have a slight section cut out and replaced with that gene of your choice. So you've now got a plasmid to carry your DNA and the DNA inserted into that. And that can be then inserted into another bacteria, which will then go on to replicate and reproduce really, really rapidly, producing many bacteria that produces a lot of insulin. So there are negatives to this. People are worried about putting genes from one organism into another and playing around with the genetics. And where these are made into genetically modified crops, like, for example, drought resistance being put into crops, people are worried that they are going into the food system and they don't know what the consequences are going to be, even though already a lot of the crops that you buy from the supermarket will be genetically modified. But there are lots of worries about it and the unknown side effects surrounding genetic engineering. Topic 7 is called ecology. It's the last topic for paper 2. It links into food chains and how organisms interact with each other. There's a required practical on sampling organisms, and then it moves through into how humans are impacting the earth and how we are affecting biodiversity. First of all, then, we'll look at the idea of competition. And firstly, we'll look at a couple of keywords that are important in this topic. The first one is population. And with population, that means all of the organisms of just one species living in a habitat. For example, you could have a population of elephants, or you could have a population of water lilies, for example. Community is the populations of different species living together. So, for example, we might describe a woodland community which comprises of a number of different species, including owls and beetles, etc. Fundamentally, whether you're talking about the individuals within a population or the individuals within a community, there is a lot of competition between those individuals. For example, in plants, plants would compete with each other for things such as light, space, water, and minerals or nutrients that they take up through their roots. Animals, on the other hand, will compete with each other for things such as territory, so similar to plants in the fact that you're talking about space, so territory or space, food, water, and mates. And this links into the evolution topic that we've looked at, whereby those that are best suited for the environment and are the best competitors will then go on to reproduce and pass on their genes to the next generation. Another important word that I want to introduce is the word interdependence. Inter is like between, and dependence dependent on each other. And this is the case where different species are dependent on one another. For example, this bird that is building this nest in this tree is dependent upon the tree to be able to build its nest. Without that tree existing within that community, that bird would have nowhere that it could roost and lay its eggs and bring up its young. So that bird would be dependent on the tree, and we call that interdependence. In this example here, you've got two pollinators pollinating this plant. So that plant is dependent on these two organisms. Another example of interdependence is dependent on them for pollination, and in reverse, the organisms here are dependent on that plant for food. So when you start building up all of the organisms within a community into a food web, you can see with interdependence that any loss of one organism within a community due to disease or predation or something like that causes massive knock-on effects to the whole community. We're now going to look at abiotic and biotic factors. Abiotic factors are anything that is non-living, and biotic factors are living factors. Just like biology is the study of living organisms, bio is anything that is living. So the non-living factors that might affect a community are things such as anything to do with weather really, so the temperature, the rainfall, or maybe humidity or something like that, the light availability, anything really to do with the weather conditions, because they are non-living factors. They could affect all the individuals living within a community. The living factors that could affect or disrupt a community are things such as disease, predators, competition, and food availability. The interaction between the biotic factors, the animals living in the environment, and abiotic factors is called the ecosystem. So the ecosystem is how the animals interact with their whole environment, including the abiotic factors as well. So that's the largest level of organisation you have, much bigger than a community or a population. Next we're going to look at adaptation, and we could be asked in the exam to either look at how plants or animals, or maybe even a fungus, is adapted to its environment. There are three levels of organisation within adaptations. First of all, we can look at structural adaptations, and in brief, this is really what the organism looks like. There's behavioural adaptations, and it's got the word behaviour in there because it's really about how the organism behaves. And functional adaptations, which are really related to anything internal inside the organism. So internal functions within an organism. So for example, if we look at this bear over here, anything structural might be things such as its colour. So the fact that it's brown could give it an advantage for camouflage within the woodland. It could be the fact that it has got large claws to catch the fish. It could be the fact that it has thick fur, for example. Anything associated with how that organism lives. Behavioural is how it behaves. So, for example, you could be looking at whether it lives in groups or solitary. So it lives on its own, it lives solitary. That's to do with its behaviour. It could be to do with how they hunt, how far they travel within a day, anything like that. And functional is anything to do with the internal features of an animal. So, for example, its metabolism, or the reproductive processes that go on inside the animal. So, for example, this bear may hibernate, and during hibernation it lowers its metabolism. So I could envisage a 6-mark question about adaptations whereby they would expect you to look at all three different types. Next we're going to look at food chains. It's quite a simple concept, but there are lots of important words involved. This is the idea that we can link organisms in a community depending on what organism eats what. For example, in this food chain we have the grass, which is then eaten by the grasshopper. which is then eaten by the mouse, and which is finally eaten by the owl. So a few key words. First of all, this grass here, and always at the beginning of the food chain you have something called a producer. This is because it produces its own food by a process called photosynthesis, where that process gets its energy from originally is the sun. So energy is transferred to the plants and they use that energy from the sun to make their own food. That is then consumed by the grasshopper. So all of these three organisms here are called consumers, but this one is the primary consumer because it is the first consumer in the chain. The mouse is what we call the secondary consumer because it is the second organism in the food chain, and the owl is called the tertiary consumer. Now, we don't have a continual line of organisms here. Within a food chain, you only tend to have 4 or 5 or maybe up to half a dozen organisms. After that, it stops. Now, the reason for that is because not all of the biomass is passed up the food chain. That means not all of the mass of each organism is passed up each food chain. Because, for example, perhaps the grasshopper can't eat all of the different parts of the plant. Perhaps there's some structures that it can't consume. The mouse, for example, might not eat all of the grasshopper. Maybe it leaves out its wings or its legs because they find those inedible. And the owl, for example, won't digest all of the mouse. It will swallow all of the mouse, but then it will bring up things like its fur and any of the hard bones that it can't digest. So by the time it gets to the end of the food chain, there is not enough energy for another organism. Energy is wasted in other ways as well, for example, through the movement of the organisms, through the fact that they produce feces, and as we said before, the fact that there are inedible, which means they can't be eaten, parts of the organism. So by the time you get to the end, there's just simply not enough energy for another organism to fit into the food chain. That's why the arrows face in this direction. They show the direction of energy transferred. So they always have to be in this direction, moving from the producers through to the consumers. A couple of extra keywords then. We would describe the owl as a predator, and we would describe, for example, the mouse as prey. Anything that's eaten is called prey, and anything that's doing the eating is called the predator. So in the same way, the grasshopper would be the prey for the mouse, and the mouse would be the predator of the grasshopper. Next, we're going to look at a required practical for paper 2, which is all about how organisms are distributed. And this required practical is all about the idea of random sampling of a population. Population being one species, if you remember from the keywords that we looked at before. For example, if you had a very large area, say 100 metres by 100 metres, you might want to estimate the number of a particular organism that is in that area. For example, the one that's often used at school is you might want to measure or estimate the number of daisies in a particular field, and you might want to compare two different fields, sunny versus shady, for example, or different fields with different pHs, perhaps different fields with different grass types as well. There's lots of things that you could look at, but the main thing is that you can't sample the whole field. It would be too time consuming and nigh on impossible to go and calculate the number of daisies in that field. What we can do is we can subsample a much smaller area of the field. It's not to scale, but for example, we could have an area that was 10 metres by 10 metres, and what you do is you divide that grid up again with string. So there we have it. We have a grid in the middle of our field, and we've divided that grid up into 100 squares. So if we were to write a method for this, or start writing a method, we would first of all write that we would measure an area 10 by 10 metres and divide this up into a grid with squares of 1 metre by 1 metre. Then still within that grid we can't sample all of the different squares within there. So what we need to do is we need to use a random number generator, and we need to use that to pick, for example, 10 or 20 squares that we are going to sample. They'll ask you about this in the exam perhaps. The more squares you sample, the more reliable your results are going to be. So we can use a random number generator. So for example, if we assigned these squares from top left to bottom right, 1 to 100, so we'd go along and say that was 1, 2, 3, 4, 5, 6, etc. We could then use our random number generator and say, for example, that it picked out 10 squares at random. We would then sample within those 10 squares, and what would we use is a grid that looks very similar to this called a quadrat. So a quadrat is a metal grid. Sometimes they are 1 metres by 1 metres, it depends on the quadrat you've got. Other quadrats are a bit smaller, perhaps 0.5 metres by 0.5 metres or something like that. So you need to be aware of how big a quadrat you're using. I'm going to suggest we're going to use a 1 metre by 1 metre quadrat, and what we do, we would then place that on one of our sampling squares, and we would count the number of daisies within that quadrat. So we can build up our method here. We've used a random number generator, and now we are going to count the number of daisies in each of the 10 squares by placing a quadrat over the grid square and counting the organisms we find, or counting the daisies in this case that we find. And we would repeat that for each of our 10 squares, and then we would take an average number of daisies per quadrat, and then we would then have an average number of daisies per metres squared. And with that information you can then compare that to a shady field, repeating it in the same way, measuring an area, dividing it up into a grid, using a random number generator to pick, for example, 10 squares, placing a quadrat on those squares and counting the number of daisies within the quadrat. The second way that you can sample organisms is by using a transect. This is slightly different, although it does use those metal grids that we used before called quadrats. It doesn't have to use quadrats, but it can do. For example, what you might want to do is look at the distribution of organisms in a straight line away from a tree. And what you would do was you'd lay down your measuring tape, which is your transect line, and you would lay that down on the ground away from the tree. Then, at regular intervals, For example, every metre or every 2 metres, you would sample the organisms. And to do that, you could either just count the number of organisms that were touching your tape measure or your piece of string that you put down as your transect line, or you could place your quadrat there and count the number of organisms that you find within your quadrat. Not only could you do that in a horizontal way, you can also do transects vertically, for example, up a tree. You could lay a piece of string or a measuring tape up a tree and sample something like leaf size or lichen cover as you go up the tree. So you might not want to use a quadrat, or you might just want to have a look at regular intervals next to that string line or tape measure. You might want to measure the size of the lichen that you find. In the same way that you can do that on a wall, for example, here you could lay a transect vertically on the wall, and next to the transect at regular intervals you could either place a small quadrat or you could simply measure the size of the lichen at that point. With a quadrat, not only might you want to count the number of organisms, but for some organisms this is particularly difficult. And instead, you might want to calculate the percentage cover in the quadrat instead. And you may well be asked to do that in the exam. So, for example, if I draw a very, very small, simple quadrat that is 5 squares by 5 squares, in total there are 25 squares. And let's say, for example, that we put this on the wall and we had a particular coverage of lichen, like so. We would count the squares that are covered with lichen. In this case, there are 1, 2, 3, 4, 5 squares that are covered in lichen, and we would use that to calculate the percentage cover. So you do the number of squares with the organism in it divided by the total number of squares, and then multiply by 100 to get your percentage. So in this case we've got 5 divided by 25 multiplied by 100, and that will give us our percentage cover of 20%. We're now going to look at the water cycle. The water cycle basically explains how on Earth the water is constantly cycled around and reused. So there's some important key words. First of all, evaporation. So this could be water evaporating from the sea, for example. That water would evaporate, and when it gets higher up in the atmosphere, it would cool down and it would condense into clouds. So we've got first evaporation and then condensation. As those clouds move around, at some point they will then turn into rainfall, and this is called precipitation, and that rain will fall back down onto the earth. At this point it might flow, for example, into rivers, straight back into the sea, or it might get taken up by plants, and when plants take up water, that water can then be evaporated from their leaves. So this comes up in paper 1 as well, this process whereby plants take up water and evaporate it from their leaves, and this is called transpiration. So in this process here, through evaporation, condensation, precipitation, flow back into the rivers, and transpiration, that is how the water is cycled on our planet. So a lot of key words there. You might see, for example, the idea that if you have some water in an iceberg, for example, on the sea, they might ask you to suggest how that water ends up in a lake. So here you would have the water, the iceberg melting into the sea, that water evaporating, condensing into clouds, and then you would have the precipitation that would lead to the rainfall filling up the lake. Further to the water cycle, you also need to know how carbon is cycled. So carbon is a compound that you're probably well aware is in the gas carbon dioxide. So carbon dioxide has a major involvement in moving this carbon around the atmosphere, but it doesn't always have to involve carbon dioxide. Let's start over here first of all. Photosynthesis is part of the carbon cycle because plants take in carbon dioxide and then they add that to water that they take up through their roots and they make a substance called glucose and oxygen. And that you'll see balanced out with sixes in front of the oxygen, the water and the carbon dioxide. So this process, it takes the carbon from the gas here and turns it into, cycles it so that it then goes into this molecule here, which is glucose. Animals will then eat the plants and therefore they will consume the carbon here from this glucose molecule and that will be taken in by the animal. Animals will then both respire and produce waste, so some of the carbon will be in the waste here, but it'll also be released by the animal as CO2 when the animal respires. Plants also respire. So the respiration from animals and plants will again release CO2 back into the atmosphere. When the animal and the plants die, the carbon that is in these organisms will be recycled through the process of decay. Microorganisms are mainly responsible for decay. So the microorganisms will feed on the dead plants and animals, and they will also respire. It's not just the larger animals and plants that you think of as respiring, but also these microorganisms are respiring, and therefore they are also releasing CO2 back into the atmosphere. Vehicle emissions are also responsible for releasing carbon dioxide. So the carbon starts off first of all locked up in fossil fuels as the hydrocarbons, and then when they are burned through a combustion reaction, that is, as we looked at in the chemistry topic, combustion results in carbon dioxide and water being released, and that is another way that carbon is cycled. And finally, factory emissions. So they are burning fossil fuels, also releasing CO2 into the atmosphere, and this completes the carbon cycle. This next section looks at how humans are impacting on Earth, and often this is in a negative way. So in recent years, we've seen a massive population rise of people, verging on the 7.5 billion people on the planet. And this has massive consequences for a number of resources such as water, land and air. The presence of these billions and millions of peoples has meant that water is regularly contaminated with sewage and other toxic chemicals. Another way water is contaminated is through agriculture. So there's a lot of use of chemicals, fertilisers on the land, and these can, what we call, run off the land into the rivers and can contaminate the water sources, therefore affecting animal and plant life within the water. We contaminate the land in a number of ways, one of the big ones being landfill, so putting all of our waste and sometimes toxic chemicals into massive areas on the land. So having toxic waste on there obviously pollutes the soils, which makes it difficult to ever be used for anything else again after the landfill. There's lots of chemicals on there. And one problem that we've had in generating more electricity is the use of nuclear fuel to do that, and we have a problem with nuclear waste and where we can store that, because wherever that is stored, that will cause massive contamination. So we pollute the air in a number of ways. When we burn fuels, we have solid particulates released. This links into chemistry topic as well, and this can lead to global dimming. We have chemicals such as sulfur dioxide and nitrogen oxides released, and they can lead to acid rain. The main problem with all of that pollution is that it is leading to a reduction in biodiversity. We said before that bio is the living things and diversity is the variety of living things that we have on our planet. The more diverse our planet is, the better it is for sustainability and future populations. However, our impact on the water, land and air ultimately is reducing that biodiversity by causing local populations of animals and plant life to become extinct. And this is the definition for biodiversity that you need to know: the variety of different species of organisms on Earth or within an ecosystem. Other major impacts that we are having are through deforestation, which is the cutting down of large amounts of trees. When these trees are cut down, it could be, for example, for agriculture, to allow crops to grow or for a space for animals to be. Could be that we're simply cutting down that wood to use it for timber. But ultimately, when a tree is growing, it is taking in CO2 from the atmosphere and it is locking that carbon dioxide up. However, when that tree is cut down and when it is burned, the burning of that tree will then release carbon dioxide back into the atmosphere. And that has repercussions because it links into global warming, which we'll talk about in detail in just a minute. And ultimately, this therefore is leading to the reduction in biodiversity, not only through releasing carbon dioxide and causing global warming, but also from the primary impact, which will be loss of habitat, which then links to the species living in that area having massive problems and sometimes locally becoming extinct within that area. I'm sure that deforestation is a concept that most of you are aware of already. However, there is something called peat bogs, which you may be less aware of. If you're a gardener, you may well have heard that word peat, and it's similar to compost, and it's something that you can use to grow your plants in. And peat bogs are acidic and waterlogged areas. And because they are acidic and waterlogged, it means that the plants that die here don't decay properly. And therefore that means that in these peat bogs you have carbon that is locked up in a similar way to having carbon locked up in these trees before you cut them down. In this peat, the carbon is locked up. However, as you can see, we're cutting away at these peat bogs, and we're taking the peat not only to use for gardeners, but also to use as a fuel as well. Because it is carbon based, it's quite a useful fuel. However, as soon as you cut into that, there becomes a problem associated with these organisms involved in decay. Because as soon as you cut into the peat bog, it comes into contact with air. And then the microorganisms can then decompose the carbon compounds that are in there, or the plants that are in there. And then whilst they are doing this, they are respiring and releasing carbon dioxide. Okay, so as soon as that comes into contact in the air, the decay process is initiated again, and that will then involve respiration, which is releasing CO2. Secondly, if we burn this fuel that we're using it for, again, we are releasing CO2. Destruction of the habitats around these peat bogs is therefore going to reduce the biodiversity again, because the loss of habitat and very particular organisms that are living in these peat bogs will therefore result in, again, a further reduction of biodiversity on our planet. We'll now move on to global warming, which is a topic that comes up in chemistry as well. So with global warming, the main gases that are involved create a layer in the atmosphere, and these gases are carbon dioxide and methane, and to a lesser extent water vapour as well. These greenhouse gases, as we call them, create a layer in the atmosphere. You won't probably have to go into this much detail in biology, however, as it goes across both subjects, it's really important to know anyway. The sun releases shortwave radiation, and that goes straight through the greenhouse gases. But when it hits the Earth, that is reflected as long-wave radiation, and this does not get through the greenhouse gases. This is re-radiated back in all directions, including back to Earth's. So it allows short-wave radiation through, but the long-wave radiation that's reflected from Earth is re-radiated. And it's because this long-wave radiation is a form of thermal radiation, this will cause the Earth to warm and heat up. So there are a number of different consequences associated with global warming, like we've talked about previously, a reduction in biodiversity. And this is to do with a number of different factors. It could be rising sea levels, and this will cause problems for humans as well. It could cause a change in organism distribution or in migratory patterns. So those animals that are able to migrate and move, they might choose to live in slightly different areas and slightly different regions because of the changing temperatures on the planet. It could cause a change in rainfall. That could be more rainfall in some areas and less in others, so it could cause droughts or severe rainfall depending on where you are, and also more extreme weather events. So after all the doom and gloom about how humans are reducing the biodiversity on Earth, a little bit of positive news on how we are trying to maintain biodiversity. So there are several programs that are put into place to protect wildlife and maintain biodiversity. Some of these are government led and other these are charities or not-for-profit organisations. One example of a program is within the countryside to maintain or reinstall hedgerows, allowing the landscape to be less fragmented and providing pathways for animals to travel more easily between different areas within their habitat. And the introduction of these field margins. And this is when farmer leaves a little bit of land set aside away from the field. So here he would have all his monoculture crops, so one individual crop, he won't want any weeds on there, he might be spraying chemicals on there, but at the side of the land just has this wild habitat available to increase the biodiversity within the farmland. So here we could have plants suitable for bees and insects and loads of other wildlife, whereas here we have our crops growing. There's linking into this, there are the major habitat regeneration programs. So, for example, money might be invested into regenerating wetlands or regenerating ancient woodlands. There's breeding programs that are linked into zoos and specialist breeding centres to try and increase and release the number of rare species in the world. So, for example, if you wanted to reintroduce water voles in particular areas, there are organisations involved in breeding those water voles and making sure they have a successful release into the wild. In general, we can all recycle, and there are major schemes encouraging us to recycle, reuse, especially with the changes in the laws of plastic bags, and reduce how much we are consuming. Finally, the government may have very particular commitments to carbon dioxide release. So they might put tariffs on very major companies to make sure that they're not releasing so much carbon dioxide into the environment. They may have incentives for them and rewards or sanctions if they do release too much carbon dioxide into the environment over the course of a year, for example. So all of these things will work to help maintain and improve the biodiversity. However, there are conflicting pressures with all of these programs that you might have to evaluate in the exam. So things such as the cost. A lot of these programs have a high cost. There is also a very close debate between the importance of biodiversity versus food security. So food security is making sure that we have enough food to feed all of the human population. And you can imagine, for example, the idea of these field margins would have a very close debate between the need for these field margins for biodiversity, but also the fact that they take away from an area where you could grow crops, so it could be affecting our food security in the future. Also, for other countries that are finding it very difficult to grow enough food to maintain their populations, well then it's very difficult to try and get governments to instill very particular commitments on those countries when they are going to want to cut down trees, to grow food, etc. And then this really leads on to the final conflicting pressure that countries can't agree. There's a lot of politics involved in setting these sanctions by the government, and very large meetings between a number of different countries to try and agree to the amount of carbon dioxide that each country is releasing. And sometimes they just don't agree because there's different levels of development in the country, and there's not an easy solution to please everybody. So that brings us to the end of the revision video for Biology Paper 2. I do hope you found it useful, and best of luck in your exam.